Hello, welcome back for the second video in our final project background series. This one we're going to cover molecular evolution of TEs. And my goal for this video is to give you the background you need to interpret graphs like this one on the left, where this is called a transposon landscape, and it tells you something about the types and when transposons were active in invading the genome. We won't get to it, but also give you, in this um, lecture, hopefully you'll see applications like the one on the right here, which comes from the Human Genome paper, where they was one of the first examples that showed that males have a higher mutation rate than females. And the way they found that was they looked at transposable element families on the X chromosome and Y chromosome that had invaded at the same time, and they saw that the Y chromosome transposons were diverging from each other at a much faster rate. And that suggested that there must be some mutational input difference between males and females. So let's talk about molecular evolution. First thing we need to understand is inferring a consensus sequence and what that can tell us about the evolution of the related sequences. So in my little cartoon example, I'm going to go through these 7 sequences, which we can pretend these got pulled out of a genome based on clustering by sequence similarity and whatever. We're going to actually run this in our software pipeline for the final projects. But these are identified as repetitive and belonging to the same transposable element type. They're sufficiently similar in sequence. So if we rewind history all the way back to the whatever organism it was at the time when this genome didn't have any copies of this transposon in it, at some point, this transposon gets in. And we won't get into the weeds about how people think this might happen. But in short, maybe the sort of evolutionary origin of most transposable elements is actually communicable viruses. So things that are just out there, uh, call them free living viruses. That's how we would talk about if it was a bacteria, but regular viruses. These are just viruses that have learned to live in the genome. So at some point, this virus gets into the genome. So the genome only contains one, and it's an active transposable element, meaning it can move around in the genome. At some point, we'll say that that active element gets a mutation that inactivates it. Okay. So in this population, we now have 2 alleles of the same element. So these things are at the same position. It's like having 2 alleles of a gene, except we've got now 2 alleles of this transposon, one that can still move and one that can't. The one that can still, the allele that can still move copies itself twice. Well, this is pretend, may copy it in real life. We'll see examples, right? Sometimes these things just explode and make tons of copies of themselves. But now, when it copies itself, in my cartoon example, it's got 3 active copies, the 2 new ones, and the original. And then it's also got an inactive allele at position 1. Over time, the original active copy is lost. So now at position 1, we only have the inactive copy. We expect that that's probably the most frequent, well, the evidence is that it's the most frequent ultimate outcome for all transposable element copies. There's always going to be selection to favor silencing the transposition of these things. So any mutation that inactivates it is good for the genome, good for the host, because now you don't have this sort of viral element loose in your genome that could be disrupting genes or landing in regulatory elements. And there's always this interplay of transposons trying to fly below the sort of genomic immune system's radar, so they can like transpose into things that are away from genes in the intergenic regions, which we talked about in the first background lecture. But you can imagine, even if they can modify their mechanisms of transposition to avoid landing in genes or regulatory regions, they still cause chromosomal breaks. A lot of, at least many of the transposons do when they transpose. And that's also potentially leading to mutation and errors in the genome. Can also lead to problems with recombination if you have lots of similar elements that are scattered around the genome, so that they lead to aberrant crossovers. So at this point, we've lost the activity of that version that's at position 1, but we've still got 2 active copies at positions 2 and 3. Fast forward in history, and you get mutations that inactivate the copies at 2 and 3. At some point, those things are segregating, just like we saw for the one at position 1, the original in transposition. These guys have made 4 more copies, and this leads now to 7 positions in the genome that have TEs. I made a little mistake here. Position 1 no longer has an active copy, so there's 1 inactive version at 1, there's an inactive and an active at 2 and 3, and then we've got 4 active copies. So in this example, at this time point, there would be 6 active copies in the genome. Again, the selection favoring silencing is probably going to lead to the fixation of those inactive copies. Here, we've gone through enough time that these guys got a mutation, but in the original one, it's picked up another mutation. So now it has 2 differences from the original invading transposon. But there are still 4 active copies in this genome. If we fast forward some more, as the number of active TEs increase, selection for mechanisms to control these things will also increase. So any mutations that are inactivating them should be favored. And I already mentioned these, at least this first point, right? Selection for it to be less harmful, right? So if it can avoid landing in exons, that's better for the transposon in terms of survival. But then the host is also trying to evolve often these epigenetic mechanisms where it can condense the chromatin in regions where there are transposons that are active so that they can't be moved. If you fast forward, we'll say to today, those selective pressures have now inactivated all the copies. So our 4 newest copies only have 1 difference. The 2 sort of middle stage evolution both have 2, and the oldest, the original invading element, has 3 mutations in it. So the key insight here, if the mutation rate is relatively constant, which is highly likely, that's not really a debated assumption, the number of mutations away from the original ancestral active copy tells us something about the order that the copies were made. So more mutations equals older. This one that is 3 mutations away is probably the original element, given that in our example where it's the only one, it has the most mutations. Okay, so now if we go back, I guess filling in the gaps there, if we make some assumptions about generation time and demographics, right, we can use the average generation time for whatever organism we're looking at, and we make an assumption that there haven't been real extreme bottlenecks in the population size. because bottlenecks will cause things to fix faster and slower, you can make inferences about how long ago. So the number of mutations, the number of substitutions tells us something about time. We won't really get preoccupied with trying to determine what's the right time. We're just going to leave it on a scale of mutational differences, substitutions, and that'll be fine. I've sort of forgot that I had left this in there because it's called a molecular clock, that if the mutation rate is constant, the generation time, you can make an assumption about how long that is, and then the population size isn't super dynamic, then you can translate the number of differences into an estimate of number of years. Okay. So they're sort of more or less direct proxies with a few assumptions. So going back a step, when we analyze the genome, all we get are the 7 sequences, right? I just walked through the history of how those 7 sequences got to be the way they are, but I've, in this picture, shuffled those things. So we don't know, just looking at a genome, what's the order that they came in. We don't even know which one is 3 mutations away, because 3 mutations away from what? That's where we have to infer the consensus sequence. So we're going to fill in each position with the most common nucleotide at that position. So for example, position 1 is an A. Position 2 is a C, right? And that one's easy because they're all C. But you can see if we go down the line, I've filled in the consensus with the most common nucleotide at each position. Oops, got these slides out of order. Fix that. Some weirdness going on with the animation there. Okay, so we've got our consensus sequence now. That consensus sequence is an inference of the sequence of the original ancestral active TE. And if you go back and look, it is in fact the same sequence as back in the original that invaded this genome to begin with. We know that this method actually works because there have been some really cool studies that build the consensus sequence of actual transposable elements in the genome, and then reconstruct that. So you make the oligonucleotide that corresponds to the consensus sequence, and then put that into cell culture, and it is an active transposable element. I've highlighted one here, the Setmar study, where they did this, and it's just I don't know. I think it's really cool. Like, it's cool, but also a little scary in the sense that you're recreating an ancient genomic virus and letting it loose in the cells. Again, some very cool studies, and there have been some with medical implications that are helping fight things like HIV. So I'll let you read about those yourselves. The point for our study, though, is that we reconstruct a consensus sequence, and then that now allows us to count up the mutations in each one of the family members or the sequences that got clustered together that are in this family. Right, so here I've counted up the number of substitutions. And again, the number of substitution tells us about the order of invasion, right? So this one that's got 3 That's probably the first insertion of this transposable element in this genome, followed by these ones with 2, followed by the most recent ones that only have 1 difference. Okay. And so here, again, don't have to put years on it in order to realize that this is a proxy for time. Got the first one, we've got things that are less recent and things that are more recent. What does it mean if the consensus sequence doesn't appear in the sampled genome, right? So we pulled these 7 sequences, none of them match the consensus. So you can pause and chew on that for a second, but what it means is none of them are active, right? If you have to have perfect conservation of the consensus, that suggests that none of the copies that are currently in the genome are actively transposing. So that's one example of a transposon. But what happens if number 3 in our cartoon example remains active? So if you get what we're saying is a mutation to C at this position still allows this transposon to be active. That's how you get the birth of a new transposon family, right? So our original sequence is still copying into positions 4 and 5. This new sequence that has a C there, you can see this whole family now is going to share the ancestral C at that position. And as it starts to proliferate in the genome, it will have its own signature of mutation where it is, these guys are all more similar to each other because they share that C, whereas over here, they all have a T at that position. And so these guys, we can differentiate the copies of this transposon family, whoops, from this transposon family. And you can still go back and estimate the number of substitutions, say something about the order. And when you do that, and if we zoom out to look at all the transposons, we can illustrate that in the form of a phylogeny. Just like you could do a phylogeny for the similarity between different organisms, you can do that for the sequence evolution of transposable element families. So what this means, as an example, if you've never spent time reading phylogenies, is the shared branches indicate shared sequence. And in this case, the branch lengths are also proportional to the number of sequence differences. So you, like, for example, these might, a branch of that length might indicate 2 mutations or something like that. That branch length being proportional to the number of differences isn't true for all methods of drawing the phylogeny. But that, if you don't know just by looking at it, typically that's explained in a figure legend. Just to sort of make it more concrete, if we look at this family of blue transposons, these guys are all more similar to each other than they are to any other transposable elements in this analysis. And we can tell that they all invaded at about the same time because they all are sort of equally related to each other. Now, maybe there's, say, 1 mutation for these bottom 3 that the other ones don't have. But for the most part, what we can say about this family is whatever mutations happened in this family at however long ago in the past corresponds to this node in the phylogeny, that thing started copying itself really quickly. Like, it made copies of itself faster than the individual copies were accumulating mutations. So it this is where we say it exploded into the genome, and then it got silenced, and then each of those sort of copies that are all over the genome now started to accumulate differences from each other at the same time. So that's how you get these burst dynamics. So you can see in all of these families, it's the same story. It invades the genome. The genome doesn't recognize this thing right away, so it doesn't have any machinery to shut it down. And so it starts making copies of itself all over the place. Once there are enough copies, the genome starts to learn, and I know I'm being anthropomorphic here, I don't want to get into the molecular machinery of how this happens, but the genome learns what this transposon looks like, and then it uses its genome defense mechanisms, these epigenetic chromatin modification pathways, to shut this thing down. And so then all of these things are silent, and they start to accumulate mutations over time. And this phylogenetic pattern where you get these bursts of transposition and TEs copying themselves leads to phylogenies like this, right? This is one that's really zoomed in on just a few families of transposons. But they're called star phylogenies, and these are characteristic of transposable elements. So, right, these purple ones, you could say that these are things when we talk about exploding into the genome, it looks like there are hundreds of copies of this thing that all basically invaded at the same time, got shut down, and then started to accumulate differences from each other. Same thing here, except on this one, right, this sort of more feather-like pattern, what this means is it was proliferating, but proliferating more like the cartoon example that I had where it made a copy of itself, but then that got a mutation, and there was an active and an inactive copy for a while, and then maybe another copy got made where they're not copying themselves fast enough that they don't accumulate differences before they get silenced. So it's not the sort of rapid expansion, right? Sort of bottom line interpretation. This is a faster expansion because everything comes back to the same origin. Whereas this is a little bit slower expansion because not all of these copies have the same common ancestor. Now, they all come down to an ancestor back here, right? But some of these copies have a more recent, like these guys all share more common ancestor up here that isn't shared at the same time with this guy. Whereas all of these copies look like they share a common ancestor at the same point. That is a more rapid expansion, more rapid invasion than this kind. Okay. And just a couple more examples. This is the like perfect star phylogeny where everything is basically equally related to each other. That is an element that just exploded into a genome. Okay, and then this one, similarly, going fast, but not as fast an explosion as this one, because there's a pattern to the divergence where they're not all coming back to the exact same point. Okay, so that's how you interpret sort of phylogeny and why we can look at transposon landscapes and interpret something about when transposons are active in genomes. So here I've got vertebrate on the left and invertebrate on the right. These are the same kinds of graphs. And all of them have this same pattern that's in the middle, where the x-axis is the divergence time. So you've got low divergence on the left, where you have sort of zero substitutions. And then on the right, that's longer ago, where I guess all of these are scaled 0 to 50. So you can imagine 50 base pairs difference. I think these are on a percent scale, but the actual absolute numbers on the X don't matter. It's just more similar. to each other on the left, less similar on the right. And so what you can see is, for example, in the human genome, what has been super active recently are the signs and the lines. Talked about that in the first lecture, where signs, the ALU elements, are the most common by copy number. Lines are the most common by base pairs. So these things are all fairly similar to each other, right? They're down here on the left where they don't have very many substitutions away from each other. We'll show a zoomed in version of human on the next slide. But just compare that, right? So that's a pretty consistent pattern in mammal history. So that, which makes sense, right? These things out here on the right-hand side, these predate the genome being a human genome. To accumulate that many differences, if we go backward in time that long, we're going backward in mammal evolutionary history. So, right, some of these mouse copies are shared, they're in the same genomic position as the ones in human, possum, platypus, especially these really old ones. Okay? So it's the same insertion. The stuff that's really recent could be unique, and I talked about in the first lecture how lots of ALU elements, in particular, there's been a whole family of ALU elements that have expanded uniquely in humans. But then if you sort of go farther phylogenetically away, you can see that DNA elements in reptiles have been way more active, and amphibians even. So the DNA elements in Western claw frog, I think this is Xenopus, they make up the majority, right? And they're still very active. You've got lots of DNA elements that have zero differentiation from each other. And then it's unfortunate that this paper has them in different colors, but you can see that there's a lot more red over here in insects. DNA elements are red. They tend to make a higher proportion of TE content in the genomes that have been looked at for the most part. With the caveat, going back to the justification for why do we need to keep studying transposable elements, that these are all based on really bad genomes. Well, maybe not really bad genomes, but certainly ones where you have hundreds, thousands, tens of thousands of scaffolds, where we know that the reason they're so chopped up in their assembly is because there's transposable elements, and those transposable elements are not captured in these analyses. Okay. And just a couple notes here again about what's the x-axis. Sometimes you'll see it as number of substitutions. I think these are percent substitutions or percent divergence. You'll also see in the analysis from our pipeline, the x-axis is something called Kimura two parameter. Kimura distance, if you think of counting up the number of substitutions is Hamming distance. We did that in some of the Python Rosalind experiments, counting up the differences between two sequences. Kimura two parameter does that, except it puts different weight on transitions versus transversion. So a transition is, say, a C to a T mutation. That is the most common type of single nucleotide mutation in genomes because it's an energetically inexpensive and often happens when the nucleotide is methylated. Okay, so getting too far in the weeds there, but just know that it's just another measure trying to get a more accurate representation of the absolute distance between two sequences. But if we just think about it as a proxy for time, the absolute value doesn't mean that much. Okay, so last slide here. Whoops. Going back to the beginning, talking about how do you interpret these transposon landscapes. So this is a zoomed in version, more recent paper for the human genome. And what you can see is the only things with very low sequence divergence are the sine ALU elements and the orange, which is the L1s. Okay. So the only active transposons that we know of in the human genome are these retrotransposons, L1 and ALU. Historically, they've also been very important for expanding our genome, because we can say that because of the abundance of them. And this is fraction of the human genome comprised by class, or so the class of the colors, and then how long ago were those things proliferating? It's the recent stuff to the old stuff. So at some point back in mammalian evolution, there were DNA transposons that were active in the mammal genome, but by the time we're anything like a human, those DNA elements are all silenced. Okay. This one on the right is from a beetle. This is the kind of output that you're going to get from the pipeline. So this is one of the summary files that you will submit for the final projects. And it's just like this, except on the top, it's like this, except it's broken down the DNA, only DNA elements in the top row, only the line elements in this one, LTR. It also produces ones that are like this, that has them all on the same plot. But the interpretation, the way you interpret it, is the same. But note that the x-axis is flipped. Okay, and I wanted to highlight that so you avoid any misinterpretation here. Right, 0 is on the far right for these, and this is default output from the pipeline. Right, I didn't flip it on purpose. Just want to make you aware that it defaults the output to be the opposite orientation for the way we usually think about time, going left to right, and what you often see in manuscripts or journal articles. But right, interpretations are the same. In this case, you can see the relative subfamilies of lines in their activity, right? L2, which is a relative of L1 that's in human, is not very active recently, but historically it has dominated the expansion of the tribolium genome. Currently, LTRs and DNA elements are pretty active, okay, because right, there are this is telling us there are lots of base pairs comprised of DNA elements and LTR, specifically gypsy elements. Okay, gypsy is just a subclass. Okay, so if you think about our phylogeny, right, you could pretend right, this is a subclass, or you could call that, that's what these subclasses are. Subfamilies. Okay, so that's it. Hopefully that gives you a good understanding of how we can interpret the molecular evolution of transposable elements.